Hello everyone and welcome back for more biotechnology. So last time we talked about taking already existing natural drugs and modifying them to hopefully make them better through combinatorial chemistry. So now we're going to spend this time talking about actually synthesizing from scratch pharmaceuticals that can either be peptides or DNA or RNA molecules. So let's start by talking about peptide synthesis. So recall from a chapter much earlier in the semester that proteins are made by stringing amino acids together and forming peptide bonds between the amino acids. There are, of course, 20 different amino acids, and we definitely had a discussion about the almost infinite number of different combinations of proteins that you can make by putting amino acids at different points along the protein structure, what we call the primary structure. So if you're thinking that we're going to be making proteins artificially, meaning that we don't have any of the cell machinery, we don't have messenger RNA, we don't have ribosomes, we don't have any of the stuff that the cell has for both transcription and translation, you're probably thinking about this the wrong way. So making proteins artificially is going to be pretty impractical because most proteins contain hundreds, maybe thousands of different amino acids, and we just can't artificially get a protein to fold correctly, not to mention that actually getting this to be an accurate result would be very, very, very difficult. But we can make peptides artificially. Peptides are not nearly as big or as long as proteins, so when we're talking about making a peptide, we're really talking about just maybe dozens or maybe 20 or 30 different amino acids. So that's not that big a deal because a peptide really doesn't contain enough amino acids to really need to fold into any particular three-dimensional shape anyway. So peptide synthesis allows us to make a short peptide chain that contains any of those 20 amino acids in any order that we want. So what you're looking at here is a big peptide synthesizer, and these bottles up here contain any of the 20 amino acids. So depending on what we tell the software or the computer that we want, the machine can take a sample of that particular amino acid and put them together to create the peptide that we desire. So what could we possibly use peptide synthesis for? Okay, so say we synthesize a peptide that we want. We have an idea of what we want. What are we going to use it for? Well, one thing that we can use it for is something that we talked about back in Chapter 9, affinity chromatography. So let's say that we have a protein that we know we want to purify from a lysate or from a crude protein extract. So let's further say that we know this protein has a very high affinity for a certain stretch of amino acids in another protein. So we know what amino acids are in that stretch in that protein. So why don't we just synthesize a peptide that corresponds to what we find in that one protein and then conjugate or stick that peptide to a binding resin? So if we then do that and we pass our crude protein extract through the column, then our protein of interest should bind to the peptide resin, and all the other proteins that we do not want should flow through. So that's a perfectly legitimate thing for us to do. Another very common application of peptide synthesis is in vaccine and antibody development. So the body will begin making antibodies when a foreign antigen is detected. Well, guess what can definitely act as an antigen? That's right, a peptide. So if we were to inject someone with a particular peptide, that person should start making antibodies against that peptide. So that can be very useful, and we'll talk a little bit more about that later. But yes, the synthesized peptide would serve as the antigen to be injected, and oftentimes for biotechnology applications, mice and rabbits or other sorts of laboratory animals can be injected with synthesized peptides so that we can harvest antibodies from their blood plasma. So for example, if you're wanting to do an ELISA or a Western blot against a particular protein, but no commercial antibody can be bought for that particular protein, then you can actually synthesize a peptide from that protein and get rabbits or mice or donkeys or whatever to generate antibodies, and then you can use that antibody once you've purified it in ELISA or Western blot or other antibody-using applications later on. And then finally, we can also talk about regulatory peptides. So it's known that some peptides can affect physiological processes by interacting with molecules in either a positive or a negative way, either by promoting certain protein activities or blocking those activities. So this is the same sort of thing as rational drug design. If we know enough about a particular disease process or biological pathway, we can actually design a peptide to interfere with or promote some sort of aspect of that pathway so that we can block the disease. So the other thing that we can do, which is very similar to peptide synthesis, is called oligonucleotide synthesis. So just as in the same way it wasn't very practical to artificially make a protein, it's not very practical to make very, very long DNA molecules. But similarly also, we can artificially create short DNA or RNA molecules called oligonucleotides. They're not very long, they're usually somewhere in the neighborhood of 50 base pairs in length. So we can use these oligonucleotides in any sort of application in which DNA or RNA complementary base pairing comes into play. Notably, PCR primers, which we're going to talk about a lot in the next chapter, chapter 13, DNA and RNA probes for things like southern and northern blots, which don't use antibodies but rather use these oligonucleotide probes. And then also something called RNA interference, which is a way of preventing the creation of a protein by preventing the creation of an mRNA transcript. Essentially, the way this works is that we can synthesize an RNA molecule, which is considered antisense, that is complementary to the messenger RNA transcript, which is called sense. And if we get complementary base pairing to occur there, the duplex RNA molecule should get destroyed by the cell. So we can actually block the creation of certain proteins by using this sort of technology here. And in this picture, you can actually see an oligonucleotide synthesizer. You can see a whole bunch of solvent bottles down here, and then you can see a variety of different nucleotide combinations in these bottles here. Notably, if we're going to make any kind of DNA or RNA molecule, either one, we're going to need all 5 nucleotides: adenine, thymine, cytosine, guanine, and uracil. Alright, so that is going to do it for this short video on peptide and oligonucleotide synthesis technologies. Thanks for watching, and I will see you next time, and we will start talking about antibody technology. See ya.